Welcome back to the course entitled Elementary Electrochemistry. In this course, in the previous class, we have started discussing about conductance and how to measure conductance, and we have discussed about specific conductance and equivalent conductance. So, in continuation with that, today I am going to discuss the method or the procedure for the determination of conductance, that is lambda of a solution of an electrolyte. So, here in this lecture, we will discuss about some of the instruments that are used, some method that is used for determination of conductance. So, while doing so, why we need to determine the specific conductance, that is capital L of a solution, which is the specific conductance of a solution is determined by measuring the resistance R of the solution taken in a suitable apparatus called the conductivity cell. This conductivity cell is a device where you will take the solution, and this device can have different shapes and the distance between the electrodes in the cell. So, one can have a conductivity cell which would look like this. And you will have two conductors on two sides, two electrodes, which are connected externally with liquid mercury kept in these tubes. And this conductivity cell has a stopcock at the bottom for draining the solution out, and you have a lid as usual, and one can take a solution inside. So, what we know is the length, that is the length and the surface area of that particular conductor, and these electrodes are made up of platinum in general. Right. So, now this a similar arrangement can be made with a long tube which is like this, where you have the electrodes here and there, and again we know the length and the area of the electrode. So, using a conductivity cell, one has to use AC or alternating current for determination of resistance R This is required to prevent any electrolysis. If you use direct current, then there will be conductance associated with electrolysis. So the things will become complicated. That is why we use alternating current to prevent electrolysis. So what we do is we make one setup of Wheatstone bridge, which I am going to draw here. Suppose you have a conductivity cell of the first kind, and it is connected to two electrodes. These two electrodes are supplied with AC current, and it is connected through the point D, which has a variable resistance done like that, R3, and it is connected to a point Q, and here you have a point P on which you have an uniform wear C, and these 2 points are further connected. And what we connect between D and the wear C is a connector, which is a headphone. So now what happens is this PQ is wear of uniform thickness, that is, the resistance of this wear increases as the position is changed. So suppose the resistance of the left hand side is R1 and the resistance of the right hand side is R2, and R3 is my variable resistance. So now we have a solution of a particular electrolyte in this conductivity cell, and we are passing AC current, alternating current, and we use a particular definite resistance at R3, which can be changed, but during the experiment we do not change it, we keep it fixed at a value R3, and with the help of this headphone we keep this variable point moving left and right, such that at one point there will not be any sound heard in the headphone, which will indicate the point of equivalence. So suppose the resistance of this solution is X. So we have 4 corresponding resistances in this Wheatstone bridge arrangement, and we using the principle of Wheatstone bridge, when we place this director, this arrow mark on the uniform wear at some point such that the left hand side resistance is R and right hand side resistance is R2, which is actually determined by the lengths L1 and L2, right, because it is uniform cross section. So one can write that X by R3 equal to R1 by R2, or simply X equal to R1 is nothing but L1, R2 is nothing but L2 into R3. So when you determine the resistance of this conductivity solution, which we assumed here as R, which is equal to X, then the corresponding conductivity lambda is nothing but 1 by R, which is equal to 1 by X. So, using this principle of Wheatstone bridge, you can determine the value of conductance of a solution that is taken in this conductivity cell. So, one can take solutions of KCl, NaCl, HCl of different concentration and determine the conductance of it. So, what we are determining is just the conductance, and this conductance is related to the concentration. So, one cannot directly compare the conductances. So, for that one has to convert the conductances into the corresponding equivalent conductance. So, what we have determined is we have obtained the value of lambda, but to know the specific conductance, that is capital L, it is necessary to determine the area of cross section of the electrodes and the distance between them, that is L and A. The ratio L by A is called the cell constant, right. So, what will be the cell constant, which is written as capital K? What will be the unit of cell constant? Unit of cell constant L, if it is measured in centimeter, area in centimeter square, that means per centimeter will be the unit of cell constant. So, now for all experimental purposes, we should know the value of L and we should also know the value of A, which essentially means we know the value of cell constant K, which is L by A, right. So, one can design these conductivity cells with a particular length distance between the two electrodes, and one can make electrodes of very accurate cross section, and we know the cross section and all that. So, using such conductivity cells, one can determine the value of L for pure KCl solutions at different concentration and at different temperature as well. So, many years ago, Kohlrausch and co-workers co-workers determined the value of L of pure KCl solutions. And here are those values with different concentration and at different temperature. So, concentration, it is equivalents per liter, temperature is in degree centigrade, that is 0 degree centigrade, 18 degree centigrade and 25 degree centigrade. What we are reporting are the values of L in mole per centimeter unit. So 0.01, if this is the concentration, then the this specific conductance L was found to be 7.751 into 10 to the power minus 4. Then at 18 degree centigrade it was 1.2227 into 10 to the power minus 3, and at 25 degree centigrade it is 1.4114 into 10 to the power minus 3. The unit is mole centimeter inverse. 0.10 normal, the value changes to 7.154 into 10 to the power minus 3, then it is 1.1192 into 10 to the power minus 2, and at 25 degree centigrade it is 1.2886 into 10 to the power minus 2, and then at 1 equivalent, that is suppose 1 normal, 6.543 into 10 to the power minus 2, 9.82 into 10 to the power minus 2, and this is 1.1173 into 10 to the power minus 1. So what we observe is that with increase in concentration, the specific conductance value L increases, and also with increase in temperature, the value of specific conductance increases. So the value of specific conductance, it is clear that depends on both temperature and concentration. And you see the specific conductance is what? It is the related conductance of these solutions with the electrodes kept at 1 centimeter apart with 1 square centimeter area. So that has been taken care of by knowing the values of L and A. If you remember, lambda equal to L into L by A. So what we have done, what they have done, they have determined the values of conductance of these solutions at different temperatures with the conductivity cell for which the L and A were known. So using this equation, the value of L has been calculated and reported. So these values of L are now corrected for the values of L and A. So to calculate these values of L, the simple expression that lambda equal to L into A by small l was used, which essentially means that L by cell constant K is used, which means K equal to capital L by the conductance of that solution, or if you write in terms of resistance, that is K into R. So what they have done is they have measured the value of the resistance of these solutions at different temperature, and with the known values of L and A, they have then calculated the value of K and easily they have calculated the value of K and determined the value of L, that is the specific conductance. So to ascertain the value of cell constant of an unknown conductivity cell, one has to use the value of L from the table and measure the resistance, say small r, of a solution of 0.1 or 0.01 molar KCl taken in that conductivity cell. So the cell constant K is nothing but L by A, which is equal to L into R, which we have shown in the previous page, that is L into R. So this value of L from table and R is determined using the Wheatstone bridge principle, right? In some places you will see that L, or it is written as Ls, which means the specific conductance. So now, as we know R, K is calculated, right? And now lambda can be then calculated using the equation lambda equal to 1000 L by C. That means one can calculate the equivalent conductance of a solution by measuring the resistance R and hence the conductance lambda and specific conductance L as well. So now when we are trying to measure the conductance of a solution and we are using water as a solvent, one has to use a very pure form of water with minimum conductance, that is very high resistance. So, how this water is produced? First is one has to distill a normal water, tap water or any water. One has to distill it at least 2 times, one time distilled, collected and redistilled and collected, and then that water is passed through ion exchange columns to remove any residual ions of carbonate, bicarbonate which may be there because of dissolution of carbon dioxide in water, so that the water that comes out of the ion exchange resins, cation exchange and anion exchange resins, should not have any ions H plus, OH minus or any other ions like carbonate or bicarbonate or dissolved sulfur dioxide which forms SO3 minus 2 minus ions. So, those ions should not be there in the water. One has to use that ultra pure water for the determination of conductance of any solution. So, before we end today's class, I would like to give you one problem to solve. The problem is at 18 degree centigrade, the resistance of 0.1 normal KCl is in a conductivity cell is 86.8 ohms, and that of 0.05 normal NaCl is 203 ohms. What is the equivalent conductance of NaCl? So, you will need the value of specific conductance of KCl solution at 18 degree centigrade from the table, and then you try to solve this problem. We will continue from here in the next class. Thank you.